All right, lipid containing stuff, molecules, proteins, that kind of stuff. We start off with phospholipids. Most commonly, the one that we talk about is the phospholipid bilayer. This is the membrane that surrounds all of our human cells. So if you ever get asked a question about membranes, we're looking for something that has to do with our phospholipids. We have a phosphate group. The phosphate group is going to be hydrophilic, so it likes water. And the fat group, which is fatty acid chains, are going to be hydrophobic, afraid of water. So they're going to be towards the inside. And so we have a hydrophilic head heading out. There's a whole row of them. And then on the opposite side, we have another one. It's going to look like this. And this makes all of our membranes. Now, steroids, as well as lipoproteins. Steroids, what we refer to as steroids, are hormones. And we're actually going to talk more about hormones in physiology, as well as the pathology section. However, the big thing to remember is that all of our hormones, all of our steroid hormones, are made from cholesterol. Cholesterol is not necessarily a bad thing. Too much cholesterol is a bad thing, but we need cholesterol in order to produce hormones, in order to produce steroids. Anytime you see cholesterol, if you see sterol, S-T-E-R-O-L, that is referring to cholesterol, all right? Just like our other fats, it is derived from acetyl-CoA. The production of steroid hormones, this is where we need to know some of the names, some AKAs, some intermediates in production of steroid hormones. We start off with acetyl-CoA. Acetyl-CoA turns into HMG-CoA. Then we have an enzyme called HMG-CoA reductase, which turns it into mevalonate. Mevalonate goes to squalene. Squalene goes to cholesterol, and the cholesterol goes to steroid hormones. So similar to some of our other questions, kind of like the amino acid type questions, I'm saying, hey, what's the precursor or what's the intermediates of these types of things? Those are the same kind of questions that we're going to get asked here. So we can also get asked kind of what is the precursor to a lot of these types of steroid hormones, such as the mineral corticoids, glucocorticoids, as well as our sex hormones. Most often, they like the answer to be either cholesterol or squalene are the ones that they like the most, but they can have any of these be potential answers. As well, we always want to know the rate limiting enzyme. So we have desmolase. Desmolase is the rate limiting enzyme specifically for steroid biosynthesis, so going from cholesterol towards the steroids. And then it's HMG-CoA reductase is the rate limiting enzyme in the formation of the cholesterol. So there's two in this whole process. Additionally, cholesterol is eliminated in the feces. Later on, we'll talk about digestion and exactly how we break up fats and do all that kind of stuff. That is in physiology section. So we'll get to that a little bit more and cover this again. But it's thanks to the bile salts, thanks to bile in general, that we're able to digest our fats, and that is produced in the liver and then concentrated and released from the gallbladder. Mixes with all of the fats and then eventually gets reabsorbed, or it can go out in the feces. Ketone bodies as well is connected to all of this, the HMG-CoA synthase. It's the rate limiting enzyme of ketone body synthesis. Most of the time, the question that we're going to ask is which of the following is a ketone body, or maybe which of the following is not a ketone body. We have beta hydroxybutyrate, acetoacetate, and acetone are our ketone bodies. Lipoproteins. Lipoproteins are exactly what they sound like. Part lipid, part protein, so they're a mix of the two, and they have very specific jobs. We talk about chylomicrons. These are secreted from the intestines, specifically the small intestines, and these are responsible for packaging and transporting the dietary fats that are absorbed by the body. And then they go into the lymphatic system in order to then get to the blood and travel through the body, mostly to the liver to be distributed to wherever they need to go. We also have VLDLs. These are very low density lipoproteins. If you've ever heard fat is less dense than muscle, so our fats, the more fat that we have, then the more fat that we have compared to protein, the less dense something is going to be. So all of these, the VLDL, LDL, HDLs, are going to be synthesized primarily by the liver and have a couple different jobs. So VLDLs as well as LDLs, their LDL stands for low density lipoprotein. Their job is to transport the triglycerides through the bloodstream and take them to all the peripheral tissues wherever they need to go. Apologies. Okay, so LDL. So the function of LDL, again, to transport triglycerides, transport cholesterol, all that kind of stuff. HDLs, these are, again, synthesized and secreted from the liver. They have a slightly different function, though. Their job is to go to all the peripheral tissues and pick up the extras. You can think of the VLDLs and the LDLs kind of like the delivery drivers. They go and they drop off all of the product, all of the fats and stuff that we need. And then the HDLs are the garbage trucks. They go and they pick up all of the waste. They pick up all the extras, package it in to bring it back to the liver. When we talk about this, LDLs and VLDLs are our quote-unquote bad lipoproteins. The HDLs are our healthy or our good lipoproteins. So looking at this, HDLs are the most dense because they have more proteins compared to fats. LDLs are the next most dense. IDLs, these are intermediate density lipoproteins, not very commonly asked, but occasionally asked. VLDLs, very low density lipoproteins, and then finally the chylomicrons. Most of your questions are going to stem from which one is good versus which ones are bad, and then where do the chylomicrons come from? And they come from the intestines. Okay. We'll go ahead and finish off the fatty stuff. So fatty acid metabolism, starting with fatty acid synthesis, also known as lipogenesis. This occurs in the cytosol, and I've mentioned this a couple of times. We use acetyl-CoA. The enzyme is acetyl-CoA carboxylase. Hit the wrong button. Acetyl-CoA carboxylase is our regulatory enzyme, stringing together everything in order to produce our fatty acids. Again, stimulated by insulin, inhibited by glucagon. If we're storing stuff, like if we're storing glucose, we're storing fat. Some of the coenzymes, we use NADPH from that HMP shunt that we talked about. And we're going to be doing reduction type reactions. For breaking down fatty acid degradation, this occurs in the mitochondria. It is known as beta oxidation. Our enzyme is hormone-sensitive lipase, and it breaks off fatty acids off of the triacylglycerides, aka triglycerides, that are stored in the fat tissue. Specifically, it breaks off an acetyl CoA, and then that acetyl CoA can go into the Krebs cycle in order to participate in in the Krebs cycle and to participate in energy production. Again, stimulated by epinephrine and then inhibited by insulin. You could also say technically glucagon and all those other ones as well are going to stimulate breaking down fats. Carnitine. Carnitine is a special protein derived from lysine, one of our ketogenic amino acids. Remember, Lucy lies to get the keys. Lysine is a ketogenic amino acid, and its job is to transport fatty acids from the cytosol into the mitochondria for beta oxidation. So it's a specific protein whose job it is to bring the fatty acids into the mitochondria. Okay, and that will wrap us up for fats. Now we're going to be talking about the urea cycle.